There is a group of people spending millions of dollars to make their lives harder. Not harder in a vague, motivational poster sense. Harder in a precise, clinically engineered, biologically specific sense. They are skipping meals on purpose. They're sitting in freezing water. They are chewing rocks. They are eating dirt. And their cardiovascular age is running a decade behind their birth certificate. These are not masochists. These are the most data-obsessed, physiologically literate people on the planet. And what they discovered is something that should disturb you about the life you are currently living. Modern comfort is killing you, not metaphorically, mechanically, at the cellular level. Your genome was constructed over millions of years of violent environmental pressure. Starvation, extreme heat, extreme cold, intense daily physical strain, microbial exposure, constant masticatory load. These were not punishments. They were the operating inputs your cellular machinery requires to function. They were the triggers for DNA repair, cellular recycling, immune calibration, bone maintenance, and mitochondrial integrity. They were the on switches. You have turned all of them off. The human species now inhabits what evolutionary biologists call a thermoneutral, calorie-abundant zoo. A perfectly temperature-regulated, abundantly stocked, biologically static cage. And by eliminating every stressor your biology was built to expect, you have silenced the survival pathways responsible for longevity, tissue repair, and metabolic resilience. The elite figured this out. Now so will you. 6 factory settings, 6 biological switches your ancestors kept permanently on, and 6 clinical protocols the most performance-obsessed humans alive are using to turn them back on. Habit 1, strategic famine. You do not have a nutrition problem, you have a timing problem. Every time you eat, you activate a serine/threonine protein kinase called mTOR. The mechanistic target of rapamycin. mTOR operates in 2 complexes, mTOR-C1, which responds to incoming amino acids, insulin, and insulin-like growth factor 1, and drives cellular growth and protein synthesis. When mTOR-C1 is active, it phosphorylates and locks down the ULK1 complex. And when ULK1 is locked down, autophagy stops. Autophagy is the mechanism by which your cells eat themselves. That sounds alarming. It is the opposite of alarming. It is how your cells destroy damaged organelles, dissolve toxic protein aggregates, and recycle molecular debris that would otherwise accumulate into dysfunction and disease. Autophagy is cellular sanitation. And when you eat every 3 to 4 hours from the moment you wake up, you never activate it. The consequence is the accumulation of senescent cells. Zombie cells. These are cells that have hit a wall, permanently arrested in their cell cycle through the activity of tumor suppressors, including p53, p21WAF1/CIP1, and p16INK4A. They cannot divide, they refuse to die, and they secrete a continuous, highly toxic cloud of inflammatory cytokines, including IL-6, and chemokines, including CXCL1, and tissue-degrading matrix metalloproteinases. This secretory pattern is called the senescence-associated secretory phenotype, the SASP. The chronic systemic inflammation it generates has a clinical name, inflammaging. It is the slow-burning cellular fire that underlies almost every major degenerative disease associated with aging. mTORC1 is required to translate SASP components, suppress mTORC1 through fasting, and SASP synthesis halts. Simultaneously, autophagic flux clears senescent cells through a process called senolysis. Longevity physician Dr. Peter Attia previously utilized 7-day water-only fasts once per quarter and 3-day fasts monthly to achieve maximal autophagic clearance. The modern clinical consensus has evolved. Research confirms that up to 2/3 of weight loss during prolonged fasts originates from lean muscle mass rather than adipose tissue. This is a severe biomechanical liability. Attia now utilizes time-restricted feeding protocols, a 16 to 8 or 14 to 10 daily eating window, punctuated by occasional 24 to 72-hour fasts. Some elites deploy rapamycin, a macrolide compound that directly binds to the FKBP12 protein to chemically inhibit mTORC1, artificially mimicking the biochemistry of starvation without sacrificing lean tissue. Tech entrepreneur Brian Johnson ran rapamycin protocols for 5 years before discontinuing due to clinical complications, transitioning instead to a precise 2,250 calorie daily ceiling with strict nutrient cycling. You are not supposed to eat constantly. Your cells are waiting for the silence. Habit 2. The soft food crisis. Here is the architectural consequence of modern food that nobody talks about. For millions of years, the human jaw processed incredibly tough, fibrous, mechanically demanding food, raw tubers, dried meat, dense plant matter. The mechanical load of heavy chewing placed persistent biomechanical stress on the maxilla and the mandible. This was not incidental, it was osteogenic. The force was transmitted through the periodontal ligaments into the alveolar bone, where osteocytes detected the mechanical strain and modulated the Rankel/OPG signaling axis. Vigorous mastication elevated osteoblast activity over osteoclasts, driving bone apposition along the facial sutures, particularly the mid-palatal suture, and expanding the jaw forward and laterally. The masseter and temporalis muscles drove healthy anterior growth rotation of the mandible. The result was wide dental arches, minimal tooth crowding, and crucially, a broad, patent, oropharyngeal and nasal airway. Then came agricultural processing. Then came industrial food. The modern diet is liquid, soft, and mechanically trivial. The jaw muscle strength required to eat it is nearly half that of ancestral populations, and without the osteogenic trigger of heavy chewing, the maxilla fails to expand laterally. Instead, it grows downward and backward, a clinical presentation called hyperdivergent growth. The palate narrows, teeth crowd. But the most serious consequence is invisible. The narrowing of the palate constricts the retropalatal airway and leaves insufficient space for the tongue at rest. The tongue falls backward into the pharynx during sleep. The airway collapses repeatedly throughout the night. The clinical term is obstructive sleep apnea, and OSA delivers a cascade of intermittent hypoxia, fragmented sleep architecture, and chronic sympathetic nervous system hyperactivation that accelerates cardiovascular aging, drives hypertension, and systematically degrades metabolic function. The biohack is to reintroduce the lost mechanical input. Elites use exceptionally hard natural mastic gum to purposefully hypertrophy the masseter and temporalis. They practice orthotropic tongue posture, pressing the entire tongue flat against the roof of the mouth, applying outward pressure to the mid-palatal suture throughout the day. For structural correction, devices like the Vivos DNA appliance and anterior growth guidance appliances place continuous multi-directional tension on maxillary sutures, stimulating osteogenic expansion. In adults with heavily ossified palatal sutures, mini-implant-assisted rapid palatal expansion, MARPE, uses micro screws anchored directly into the palatal bone to physically separate the maxilla. Clinical data shows this increases nasal cavity volume by up to 29%, dramatically reduces nasal airway resistance, and resolves sleep-disordered breathing at its anatomical root. Your ancestors had wide jaws because they used them. You have a narrow airway because your food was processed for you. Habit 3. Thermal hormesis. You live at 22 degrees Celsius every hour of every day. Your ancestors lived nowhere near 22 degrees Celsius. They endured the midday heat of equatorial Africa and the sub-zero nights of Eurasian glacial epochs. Not as an inconvenience, as the mandatory trigger for two of the most powerful molecular repair systems in the human body. When core body temperature rises above 38.5 degrees Celsius, intracellular proteins begin to denature and unfold. These misfolded proteins bind competitively to the chaperone HSP90, liberating heat shock factor 1. Free HSF1 trimerizes, translocates into the nucleus, and binds to heat shock elements on the genome. This exponentially upregulates the HSP70 and HSP90 chaperone families. These proteins refold damaged molecules, dissolve toxic aggregates, and direct irreparably damaged proteins to the ubiquitin proteasome system for degradation. Intracellularly, HSP70 sequesters pro-inflammatory mediators and downregulates NEF-kappa B, the master transcription factor of the inflammatory cytokine cascade. HSP90 stabilizes and correctly positions the proteins involved in DNA double-strand break repair, including BRCA1, BRCA2, CHK1, and the MRE11/RAD50/NBN complex. Extracellularly, HSP70 activates immunosuppressive regulatory T cells through MHC class II presentation, further blunting systemic inflammation. Cold operates on an entirely different molecular channel. Peripheral cooling activates transient receptor potential channels, triggering a massive sympathetic catecholamine surge that elevates norepinephrine by up to 300%. Norepinephrine binds to beta-3 adrenergic receptors on brown adipose tissue. BAT is mitochondria dense and expresses uncoupling protein 1, UCP1, which physically decouples the mitochondrial electron transport chain from ATP synthesis, dissipating energy directly as heat. This non-shivering thermogenesis clears triglycerides and glucose from the bloodstream and fundamentally improves insulin sensitivity. Simultaneously, cold upregulates RNA-binding motif protein 3, RSBM3, and cold-inducible RNA-binding protein, CIRP. RBM3 stabilizes mRNAs, drives synaptic regeneration, prevents neuronal apoptosis, and has been shown in murine models of Alzheimer's disease and prion disease to halt neuronal loss, rescue cognitive function, and extend survival by reversing synaptic pruning. The Elite Protocol. Minimum 57 minutes of sauna per week at 80 to 100 degrees Celsius with 10 to 20% relative humidity, divided into multiple brief sessions. Cold water immersion at 10 to 15 degrees Celsius for 11 minutes per week, divided into 1 to 3 minute plunges. Cold is applied early in the day to leverage the catecholamine spike for cognitive performance. Heat is applied in the evening to allow the subsequent drop in core body temperature to facilitate deep sleep architecture. You are paying a biological price for every day you spend at a comfortable temperature. Habit 4. The dirty immune system. Modern hygiene is not protecting your immune system. It is leaving it completely untrained and chronically overreactive. The Old Friends hypothesis proposes that the mammalian immune system co-evolved over 500 million years in continuous intimate contact with harmless environmental microorganisms, soil bacteria, and tolerated helminths. Your immune system was not designed to regulate itself in a vacuum. It was designed to use diverse microbial data as calibration input. Without this input, it defaults to a permanently aggressive, pro-inflammatory state. The mechanism is specific. When the immune system encounters signal molecules from harmless soil-based organisms, tolerogenic dendritic cells and the mucosal epithelium detect these signals through pattern recognition receptors, specifically toll-like receptors. This activates PI3K/AKT and mTORC1 signaling pathways within the immune cells and drives the secretion of anti-inflammatory cytokines, primarily interleukin-10 and transforming growth factor beta. Critically, this cascade drives the expansion, differentiation, and maturation of regulatory T cells. TREGs are the immunological brakes. They suppress hyperactive effector T cell responses along the Th1, Th2, and Th17 pathways that attack host tissue in autoimmunity or overreact to harmless proteins in allergy. Without the continuous microbial data from soil and environmental organisms, TREG populations fail to expand. The immune system remains on high alert indefinitely. The epidemiological result is visible. The exponential rise of type 1 diabetes, multiple sclerosis, inflammatory bowel disease, and severe allergy tracks almost perfectly with the sanitization of the urban environment. The biohack is strategic re-exposure. Standard dairy-derived probiotics like generic lactobacillus strains typically fail to survive gastric acid and do not replicate the diversity of ancestral environmental exposure. Longevity protocols have shifted to soil-based organism probiotics. Spore-forming bacteria endemic to soil, specifically Bacillus coagulans and Bacillus subtilis, with their thick-walled spores that survive digestion. Clinical data confirms these spores engage epithelial pattern recognition receptors, reduce intestinal permeability by lowering zonulin, suppress pro-inflammatory chemokines, including interleukin-8, and repair tight junction integrity. Elites supplement with SBOs. Consume unwashed organic produce from trusted biodynamic farms, and engage in deliberate grounding and exposure to biodiverse natural ecosystems. They track baseline inflammation via high-sensitivity CRP. Your immune system is not overreacting, it is underinformed. Give it back its data. Habit 5. The light dictator. Light is not an aesthetic condition. Light is a hormone. The master circadian pacemaker of the human body is the suprachiasmatic nucleus, a cluster of neurons in the anterior hypothalamus. It receives photic information exclusively from intrinsically photosensitive retinal ganglion cells, iPRGCs, which express the photopigment melanopsin. Melanopsin is maximally sensitive to short-wavelength blue light in the 415 to 480 nanometer range. When morning sunlight strikes the retina, iPRGCs transmit a direct signal via the retinohypothalamic tract to the SCN. The SCN immediately activates the hypothalamic pituitary adrenal axis and the autonomic nervous system, triggering a sharp elevation in pituitary adrenocorticotropic hormone, ACTH, and a rapid, robust spike in plasma cortisol from the adrenal glands within 30 to 60 minutes. This physiological cortisol awakening response mobilizes hepatic glucose, raises core body temperature, and optimizes mitochondrial oxidative phosphorylation to meet the energetic demands of the active day. In darkness, the absolute absence of blue light allows the SCN to signal the pineal gland to synthesize and secrete melatonin. The public understands melatonin as a sleep drug. This is a severe underestimation of its function. Melatonin is a potent and structurally unique subcellular antioxidant. Circulating melatonin concentrates directly within the mitochondria, possessing up to 10 times the radical scavenging capacity of vitamin C. Inside the mitochondrial matrix, melatonin scavenges lethal free radicals and protects cardiolipin, the unique phospholipid of the inner mitochondrial membrane that is absolutely necessary for the structural integrity and optimal electron flow of the electron transport chain. Now understand what your phone screen is doing. Chronic artificial blue light exposure at night falsely signals the SCN that it is midday. Melatonin synthesis is completely suppressed. And without melatonin protecting the mitochondria, reactive oxygen species leak freely. Clinical assays using Seahorse XF bioanalyzers demonstrate that blue light doses as low as 50 joules per square centimeter significantly increase superoxide anion production, reduce mitochondrial oxygen consumption rates, and nearly abolish ATP synthesis. Prolonged exposure increases fission proteins DRP1 and BAX while decreasing survival proteins OPA1 and BCL2, driving the cell toward apoptosis and accelerating tissue aging at a mitochondrial level. To actively repair this, longevity elites deploy photobiomodulation, red light at 630 to 660 nanometers, and near-infrared light at 810 to 830 nanometers penetrate deep into dermal and subdermal tissues and are directly absorbed by cytochrome C oxidase, complex 4 of the electron transport chain. This targeted photon absorption dissociates inhibitory nitric oxide from the enzyme, restoring optimal electron transport and driving abundant ATP synthesis. Post-sunset, strict blue light elimination is enforced. Amber and red ambient lighting only, blue blocking glasses in the hours before sleep. Morning sunlight viewing of 10 to 30 minutes immediately upon waking, or 10,000 lux full-spectrum daylight lamps as a substitute. Your phone screen at night is destroying your mitochondria. This is not a metaphor. It is documented, measurable, molecular damage. Habit 6. The sedentary gym trap. You sit for 14 hours, you go to the gym for 1, and you believe this makes you healthy. It does not. The human musculoskeletal system was designed for continuous, low-level mechanical loading. Ancestral humans walked 10 to 15 kilometers daily, navigated uneven terrain, carried loads, and squatted for extended periods. This incessant, low-intensity movement generated what physiologists call non-exercise activity thermogenesis, NEAT, and it completely dominated ancestral energy expenditure. But beyond calories, continuous movement is the mandatory biological driver of connective tissue integrity. The fascial system is a three-dimensional extracellular matrix of collagen and a viscous ground substance. Embedded within fascial layers are fasciocytes, specialized cells that synthesize hyaluronic acid. The structural behavior of hyaluronin is entirely dependent on mechanical loading. Continuous low-level shear stress generated by NEAT activates stretch-sensitive calcium channels on fasciocyte membranes, including PSO1, TRPV4, and P2Y2. The calcium influx triggers the HAS2 enzyme to synthesize hyaluronin in a depolymerized high molecular weight state. High molecular weight HA acts as a superior biological lubricant, binding to CD44 receptors and maintaining the frictionless sliding of muscles, nerves, and joints. When you sit for 14 hours, HA molecules superaggregate and entangle. The ECM becomes a hyperviscous, glue-like structure. This pathological state is called fascial densification. The entangled HA chains break down into low molecular weight fragments that bind to RHAMM receptors, directly catalyzing localized inflammatory cascades, fibrosis, and chronic myofascial pain. 1 hour of gym exercise cannot reverse this. Only continuous, low-level shear forces throughout the day maintain the hydrodynamic properties of the fascial planes. The bone problem runs parallel. Skeletal remodeling follows Wolff's law. Bone density is maintained by continuous mechanical loading. The micro-deformation caused by simply walking and standing generates fluid shear stress within the osteocyte lacuno-canalicular network. Osteocytes respond by downregulating sclerostin and DKK1-2 potent catabolic osteokines that inhibit the WNT/beta-catenin bone-building pathway. Suppress sclerostin through continuous movement, and osteoblastogenesis runs freely. Sit for 15 hours, and sclerostin elevation overrides an hour of weight training, and bone mineral density gradually erodes. The elite solution is engineered neat. Treadmill desks running at a continuous quiet pace of 1.5 to 2.0 miles per hour during standard office work. Hours of continuous stepping that prevents HA superaggregation, suppresses sclerostin, and spikes brain-derived neurotrophic factor for sustained cognitive performance. The gym builds strength. Continuous movement maintains the structural biology that makes the body viable. Sitting is not rest. It is slow structural degradation. Here is the conclusion. You have been sold the idea that comfort is the reward for a successful life. A perfectly climate-controlled home, processed food available every 2 hours, a chair at work and a couch at home, sanitized surfaces, screen light at midnight, a soft jaw that never has to work. Every single one of those comforts is silencing a molecular switch your genome has been waiting millions of years to flip. The most expensive biological intervention available today is not a drug or a surgery. It is the deliberate, precisely engineered reintroduction of the hardship your biology was built to expect. And the people spending millions to do exactly that are outrunning their own biological clocks in ways that are now measurable on DEXA scans and mitochondrial assays and inflammatory biomarker panels. The ultimate luxury is not comfort. The ultimate luxury is engineered hardship. 6 factory settings, 6 biological switches, strategic famine, masticatory load, thermal stress, microbial diversity, optical anchoring, continuous movement. The question is which one you restore first. Subscribe if you want to go further.